instructions. So welcome everyone to the first MSA Journal Club. This is very exciting. So in the past, we've done a few workshops. Last year, Prof. Prinsloo and his group at UNISA did some wonderful NMR workshops, and we had Prof. Edwin from Venda University to LCMS workshops. But we felt these workshops a bit sporadic. We want to keep some more communications going on within MSA. So we're starting up this journal club. And for this week and next week, we're doing a very basic introduction to the main platforms in metabolomics. Most of you know these platforms, but some of you are still quite new, so we want to be able to cover all the bases before we get to the more complicated stuff. In the future, we'll have some guest speakers more focused on journal clubs, and at least once a month, we want to have a spotlight on student projects. We'll go from institution to institution, get some representatives so that students can share their projects with the group. And the idea is to have this journal club by students for students so that people can communicate. And we also want to start a forum where everyone can communicate. So to kick the whole journal club off, we have NMR as an introductory course. Prof. Prinsloo, who is from UNISA University, I think it's in Pretoria, the main campus. He is the guy who did the workshop last year, as I said. He will go through the first half on the theory background, give you the very basics of the theory, how to understand NMR, and then I'll jump in after that. Some of the practical parts, the sample preparation, and just reiterate some of the things that Prof. Prinsloo goes through. And hopefully by the end of this, everyone will have a basic idea of NMR. You're free to ask some questions afterwards. We'll only be an hour-long session every week at Thursday at 11 o'clock, an hour-long session. But for those that have other questions later on, we will invite you to join our journal club. We'll post that link later, our forum for the journal club, and you can go on there and communicate with everyone. So, Prof. Prinsloo, would you like to take it away?
Thanks, Shayne. Good morning, everyone. Yes, so we'll just start with, I'm going to handle the first part just on the basics of NMR, and it will be a bit focused on the practical application of metabolomics, NMR and metabolomics. So it's not completely just a theoretical kind of presentation. It is specifically focused on what you're going to need and what you have to look at when you're going to use NMR and metabolomics. So I'll just quickly share my screen here. Is it visible to everyone?
Not yet.
Let's try again, yeah. That's better now.
Oh, there we go. Great.
Okay, good. Okay, so let's start. This first one is just a bit of text on what NMR is, what you have to expect when you look at NMR. So the principle behind the NMR is that the nuclei have spin, so they're spinning around and they're electrically charged. And that's important as you have to put it in a magnetic field. So when you have put it in this magnetic field, then you have an energy transfer. So the energy transfer is happening, it's going to a high energy level, and then when it returns to its base level, it is emitting a frequency, and that frequency is then picked up. And that's the basis of what you're going to work with here. And then that signal is then matched with a specific area on your NMR profile. And it's related to how your for instance, in your proton NMR, the environment of your NMR, and in what state your proton is. So basically, your proton is spinning. You apply a magnetic field. You have an energy level that's it's energy transfer. So you have a high energy level. It's losing that energy. It's going back to its base level, and that frequency that is then released is then picked up by the detector, and that is what you find on your NMR spectrum. So I'm going to focus on basically 4 things here. So first, we're just going to look at the spin and how you get your signal. Then the chemical shift and splitting, which Shayne will also share with you, because that is really important. You have to understand those basic things when you want to understand what you're looking at and what you want to interpret your NMR profile. And then second, and they're just going to touch on 2D. It's a bit advanced at this stage, but that you just know what it is, or that it is available if you see that what they're talking about. But I'm not going to focus on that. It's just for information purposes. So this is what an NMR looks like. So it's just a big casing, and your magnets are inside. So you have your sample in your NMR tube that looks like this. So you put it there at the top, and it's then pulled into your NMR, and the magnetic fields are then around your sample. So, and then it's processed, and you get your NMR spectrum, and it's then just based on your magnetic field that's applied and how your protons then react. In the case of proton NMR, how your protons react to your magnetic field. So if we are then looking at how the spin and what your nuclei are doing, so your nuclei are positively charged, and they create a tiny magnetic field. So that is important as you want to put it in the magnetic field, and you want to orientate the proteins or your nuclei in this magnetic field. And it's determined by the number of protons and neutrons, your nuclei. And it's important that you need to have an odd number. So that's why we have just specific ones that we can use for NMR. So the most common ones is your proton and your carbon-13. So it's 1H NMR and your carbon-13. And it's because you need that odd number to be able to produce an NMR profile. So you can see your nuclei are spinning, and your electrons are around it, and that is going to affect your magnetic field at the end and the signals that you obtain. So this is where you have your normal the nuclei that's spinning around. When you apply a magnetic field, they orientate in a specific direction, and it's based on this small magnetic field that, because of the charge of the atoms. And what you have then, you have applied magnetic field. It's then taking up this high energy level, and the moment it loses that energy level, it will then emit that frequency, and that will be picked up. So they must be in this magnetic field to be able to pick up that frequency that's from the high energy level to the lower energy level. So 3 things that are important, that's when you look at your magnetic, you look at your NMR, and when you want to get proper analysis. So the size of a magnetic field is important. So the bigger the magnetic field, the better analysis you can get, the more resolution you have. So it's not to say you cannot use a 200 megahertz, you can use it. The resolution will just be better when you use a 600, 800, or even bigger. So the duration of your sample that's exposed in your magnetic field is also important. So the shorter your duration is, the more samples you can run. So if you have a high magnetic field, then you have just a small number of scans you can run, short time, to get a good analysis. If you do not have a bigger magnetic field, magnetic size of the magnets, sorry, then you will use a longer exposure time, so that you can have a better resolution. So, and the third one, which we're going to look at later on now, is the environment of your proton atom. and the electrons around it, the atoms that are next to your hydrogen atom. It's all important to be able to know or to see what and understand and interpret your NMR profile at the end. So the first thing we are going to look at is your chemical shift. So different protons will give rise to slightly different frequencies. And that is what you will see on your NMR profile, is that slight differences that you have between your different protons. And we talk about shielding. And it's shielded from your magnetic field. So these atoms are in your magnetic field, and you have shielding and deshielding. So the moment that proton is not shielded anymore by electrons, then you have a deshielding, and you have like different things like your oxygens that are bigger than your protons, and there are more electrons. So these kind of things will influence your proton. So this is an example. So this is what you will have. You will have your scale on your ppm frequencies. That's from 0 to 10 or 11 or 12. And this is where your peaks will be. So the peaks that you find in this region here, we call them shielded and upfield. And the lower down you go here to this side, you call them deshielded or downfield. So that's when your electrons are not around your protons anymore, and they are deshielded. Here's where your protons are around them. So this is an example. So you have two carbons, and you have your protons around it, and there's nothing here that is influencing the electrons around your protons. So these ones are shielded. The moment you, for instance, have things like oxygen or double bonds or other elements like a nitrogen or so on, then you have a definite environment here. So this oxygen is having an effect on the electrons around your protons, and that then will result in a deshielding. So when the electrons are not properly around this hydrogen, and this is then affecting how it reacts in your magnetic field, and that energy transfer, that energy level that it's taking up, and when it releases that energy will be different in this case than what you have here. And then you get peaks on this profile here. So the ones that are shielded to this side, and the ones that are deshielded more to that side. So this is something that you'll find in every textbook if you look at NMR. So this is just a general scheme of what you can expect where. This is not cast in stone, but this gives you an idea if you look at your profile, what you're going to find. So here are your, just for instance, your alkyls, your chains, single bond change, or for instance, a ring structure, but only single bonds with hydrogens and carbons. And they're normally around this region in between 1 and 2. So, and then you can see the more you have other things close to your hydrogen, it will then be deshielded in a certain way. So your nitrogen, your oxygens have an effect on them. If you have things like your chlorine or bromine, or fluorine, they will have an effect. Your triple bonds, or if you have a carbon double bond oxygen, when you have a double bond next to your hydrogens, that will affect it. And then these ones having a huge effect on your shielding of your hydrogens. So when you have aromatic rings, so for instance, in this ring, we'll have a look at this later on as well, but the electrons are moving in this field, in this aromatic ring, and it's then completely deshielding your hydrogens. So they are completely exposed. And you have then specific peaks here in this region that we call the aromatic region, which are linked to hydrogens that's on a aromatic ring like that. So this is just, for instance, your atom, and it's spinning. And the moment you then have things like your aromatic ring, so these are double bonds there. So you have electrons moving in here, and they form like almost like a cloud, an electron cloud on top and a cloud at the bottom, and your hydrogens then outside here are completely exposed. And this is then why these ones are de-shielded, and I move completely downfield to a region of 6 or 7. And this is just an example of what happens, and to get an idea or a picture in your head of what's happening to your hydrogens when they're in this magnetic field. So this is an example of a NMR profile or analysis, a proton NMR. So you have your ppms from 0 to 11 or 12, as I showed before. So normally we use a standard or a compound that is at 0, and then your samples are all aligned, can be aligned because of this standard that you applied. And then I've put this diagram here at the top so you can just compare. So if you then have peaks in this region around 1 or 2, there are no double bonds, there are no oxygens, there are no things around them. It's just single bonds or ring structures where there are no double bonds or oxygens or anything close to the hydrogens. So they are completely shielded. And then as you then move downwards, you'll then find these kind of things around your hydrogens. So this we call them, for instance, the sugar region between 3 and 4. We have your oxygens around them. You have double bonds. You have things like your double bond oxygens. And you can see here, for instance, this is a plant sample. So there are many sugar peaks here. So these are the region where you have your double bonds. And then you can see in this case, you have quite a lot of aromatics here. So between 7 and 8, you have your aromatic peaks, and these then are peak hydrogens that are situated on a ring structure like that. As you can see in this case, there are quite a few peaks there as well. So this just gives you an idea of what you have in your sample, just by looking at where your shift is, your chemical shifts. So this is important to know, to distinguish from what we're going to look at next when we look at the splitting patterns. But this is your chemical shift, and it's based on the environment around your hydrogen. Is it shielded or is it de-shielded? So it's either upfield, or you get then your peaks in your downfield. And this is then based on what is around your hydrogens on your structure or your sample or your compound. So when we look at spin-spin splitting, so this is completely different from what you find in the chemical shift. So this is the second aspect we're just going to look at. So spin-spin splitting, normally observed between nuclei that are 1, 2, and 3 bonds away. And you get chemical equivalent and chemically non-equivalent. And that is important when you're going to interpret your analysis. So for instance, in this case, you have a carbon, a chlorine, and you have 3 hydrogens here. So all 3 of these hydrogens are in the same environment. So we call them chemically equivalent. And they're going to absorb at the same, they're going to show you a peak exactly the same place. And they're not going to influence each other because they are exactly in the same chemical environment. If you look at this, for instance, your hydrogens that are attached to this carbon is next to an oxygen. So 1, 2 bonds away. And in this case, your hydrogens are next to a chlorine, 1, 2 bonds away. So the hydrogens in this case is not the same or not in the same chemical environment than those 2. So in this case, we have non-equivalent hydrogens. And this is important to distinguish between these, where these ones are exactly in the same chemical environment, where these ones are exposed to different chemical environments, and they will then be showing splitting patterns. And this is what you will find. So for instance, a hydrogen, where there are no other hydrogens around, or hydrogens that are not chemically not the same, you will have a singlet. So just one big peak. If you have a hydrogen and there's a hydrogen next to it within 2 or 3 bonds, which is in a different chemical environment, you'll have a splitting pattern. And in this way, so we call it a doublet. So these 2 peaks belong to the same hydrogen, but it is in this case, a doublet. When you have 2 hydrogens next to your hydrogen that you are looking at, you will have a triplet. And it's in this kind of formation, 2 smaller peaks and 1 bigger one in the middle. When you have 3, it's a quartet, and it goes on. So if you have more than 3 hydrogens, you, for instance, have a multiplet. But this is what you will find then on your profile. And just to highlight again, this is not the same as your chemical shift that we looked at previously. This is how the peak will look like when you have hydrogens in the environment around your hydrogen that you're looking at. So this is an example, and I know Shayne will also have a look at this, but it's important. This is really important to distinguish between your splitting pattern and your chemical shift. So this is, for instance, a structure, compound. We have a chlorine, a carbon with 1 hydrogen, and a carbon with 3 hydrogens on there. So this carbon is right next to a chlorine. So this hydrogen and those hydrogens are in completely different chemical environments. So they are not chemically equivalent. So in this case, this one is further away from your chlorine, so they will not be de-shielded, so they will be more shielded, and you expect it then closer to 1 or 2, because they are shielded. The hydrogens are not affected that much by your chlorine. So those 3 are in the same chemical environment, so they will all give a peak at exactly the same place. But next to that, those 3 hydrogens, you have 1 hydrogen that is not chemically equivalent, and that then will then give a splitting pattern of these hydrogens. And because there's only 1 hydrogen there, you get then a doublet. So all 3 of these hydrogens are then split into a doublet, and they give a peak at exactly the same place, right here. This hydrogen here is next to the chlorine, so this one will affect your shielding. So this one is more de-shielded because of this chlorine here next to your hydrogen. But these 3 hydrogens that is next to this one is, as I mentioned earlier, in a chemically different environment, so it's not chemically equivalent. So these 3 hydrogens will affect or will create a splitting pattern on that hydrogen. So this hydrogen is de-shielded, so it's going to move downfield, in this case around 6. And because there are 3 hydrogens next to it, it gives a splitting pattern, in this case a quartet. So you have your 2 smaller peaks on the side, and the bigger ones on the middle. So when you have a quartet, you know there are 3 peaks, or you have 3 hydrogens that was responsible for that splitting pattern. And when you have, in this case, just 1 hydrogen, and next to these ones, you just have a doublet. Distinguish between the splitting pattern here, which is doublet and a quartet, and your chemical shift, which is the ppms here. So this one is at 2 because of the shielding or the de-shielding, and in this case it's at 6 because of the de-shielding. Okay, so it's 2 different things. You have your splitting pattern, and you have the place where it's on this scale is at 2 because of the de-shielding, which shielded, and in this case it's de-shielded. Just one example more. So this is a bit more complex one. And when you look at your structure, it's important to look at, for instance, you have hydrogens here, carbons. So you have hydrogens on this side of a carbon attached to where the oxygen is attached to the carbon with a double bond. There you have an oxygen directly next to your carbon, and these hydrogens are attached to a carbon, just to a carbon, and that one is attached to your oxygen. So in this case, you have 3 chemically different environments. So these ones, these 3 have the same chemical environment. These 2 will have a different one because it's next to the oxygen. And these 3 have a chemical different environment because it's attached to a carbon, attached to an oxygen, and on this side also an oxygen. So you have 3 groups of hydrogens here. And because there are oxygens in this case in this structure, you will have then a different chemical shift of each of these hydrogen groups. So if you look at these ones, they are, of all these hydrogens, they are the most shielded. There's nothing next to them that is affecting this, this shielding of your hydrogens. So in this case, your chemical shift is around 1. And in this case, you have a triplet because next to each of these hydrogens, you have 2 hydrogens that is in a different chemical environment. So each of these hydrogens will then split into a triplet because of the 2 hydrogens next to it. The red ones, these 3 are chemically equivalent, but there are no hydrogens next to each. If you look at 1, 2, 3, 4, 5 bonds. So in the 2 or 3 bonds next to this hydrogen, there are no hydrogens. Next to this hydrogen, there are no hydrogens close to it that can split these hydrogens because they are too far. So it's 1, 2, 3, 4, 5 bonds to the closest hydrogen, which is too far. So in this case, your hydrogens will all give a singlet because there are no hydrogens next to it that can create a splitting pattern. And that's what you see here. Note the chemical shift is different because it's next to the carbon and these oxygens. So there's a different chemical shift, but the splitting pattern is a singlet because there are no hydrogens next to it that is influencing these hydrogens. In this case, we'll see it's a quartet because each of these hydrogens will be influenced by these hydrogens next to it. So there are 3. And when you have 3 hydrogens, that is next to these hydrogens, 1, 2, 3 bonds. So it's close enough. Then you will have a splitting pattern. And in this case, because of the 3, you have a quartet. Once again, just look at the splitting pattern of these hydrogens. There are 2 next to it, so you get a triplet. In this case, there are no hydrogens next to it, so you don't get the splitting pattern, so it's a singlet. In this case, you have 2 hydrogens. Next to it, there are 3 hydrogens, so it will be splitting in a quartet. In this case, your hydrogens are not de-shielded, they are shielded, so you have an upfield. Place where so it's upfield, the peaks is around 1, the peaks are around 1. In this case, it's influenced by all these oxygens around them. So it's completely, or not completely, but a lot of de-shielding here of those hydrogens. So it's moving down to 4. And these ones are affected by your oxygens. So you have a little bit of de-shielding and it's moving down to 2. So once again, your splitting pattern, which tells you something about your environment around your hydrogens. And then you have a chemical shift on where you find your peaks on this analysis. I'm just briefly going to mention the coupling constant because that's you can also use later on in your interpretation. So the coupling constant, which I call is J, is in frequency units and is a measure of the interaction between your pair of protons. So if you just look at this, so this the distance here we call the coupling constant, and you can measure that, and that is what we're going to look at here. So if you have your singlet, there are no J constants, no coupling constants. If you have, for instance, a doublet, the distance between this you can measure, and this is your coupling constant J for that doublet. If you have a triplet, this J value here and this J value here will be the same. So then you know it's not 3 different peaks, but they all belong to one hydrogen because of the same J values here, and the same then when you have it in a quartet, this will all be the same. So the distance here is what you measure to get your J coupling constant, and this will be the same as this and the same as this for this splitting pattern here. So I will just briefly mention 2D NMR. So the previous ones that we looked at is your 1D NMR, your one-dimensional, and you get different ones. These are just examples, but it's when you have your NMR profile and you have a two-dimensional one where you look at the interaction of hydrogens, and you have your NMR profile on this side as well. So, and then you have your spots here where your different hydrogens are interacting. So this one that is at a ppm of around 1 is close to the hydrogens that are at 4, and then you get that spot there. And the same in this case, this hydrogen that is absorbing at 7, or that's showing a peak at 7, is very close to the peaks or the hydrogen that is around 2. So that later on can tell you something about your structure, tells you something of how close these hydrogens are next to each other, and we call that in 2D NMR, and that can help a lot later on if you are not sure about this compound or a structure and you want to confirm identity, then you will start looking at 2D NMRs. So I'm not going to go into more detail here because this can get very complex. And at this stage, this is not very important for you. Most important for you is to be able to interpret a 1D NMR. But if you see 2D NMR, this is what they are talking about. Okay, so in general, so NMR is very versatile, and that is why people sometimes prefer to use an NMR, because it's not influenced, for instance, by the temperature. So an NMR profile that is generated here in South Africa and Gauteng will be the same as an NMR profile that is generated in China. So because it's looking at the chemical environment of your hydrogens. So if you use a 800 megahertz here in South Africa and you use an 800 megahertz in China and you use the same sample, you will get this probably, or you should get the same profile. It's not very sensitive, so that's one of the drawbacks of NMR. Your GCMS and your LCMS are much more sensitive, so the application there is sometimes better in some of your samples. This is not always negative, because if it's not that sensitive, it means that you will, in your samples, probably look at your differences that are compounds that are in a higher concentration. So the major compounds and the difference in major compounds. So it's not always negative, but if you look at low concentration compounds, NMR is not that sensitive, and you might lose some of your information. Important is your magnet size. So also when you analyze your samples, if you're going to use different magnet sizes, you will get different profiles and you will get different resolution on them. So make sure you always use the same magnet size when you run your samples. And the number of scans are important, and that can improve your resolution of your samples, of your peaks. And we talk about the noise ratio, sample to noise ratio. And for instance, this is what you can see here. So in this case, you can see there's a lot of noise here. There's less. In this case, there's almost no noise. And that helps you when you look at your peaks, especially when you have lower peaks. So the bigger your magnet is and the higher number of scans, you can improve your noise ratio, and you get then improvement on your peaks and your resolution of your peaks. Another nice feature of your NMR is that it is quantitative, so you get an idea of your concentration of your peaks or your compounds, which is a very nice feature of NMR that can contribute a lot of information if you look at a sample. So the higher the peak, the higher the concentration. This is in contrast to LC and GC, where you do not really have that link to your concentration. A higher peak in LC, for instance, is not to say it's a high concentration. It there depends on your detector. So in this case, in NMR, if you have a higher peak than in another sample where the peak is lower, it means that the concentration is lower. And then by using, for instance, your TSP or a standard, you can then quantify your compounds in your sample. So that is a very nice feature of your NMR. So just to finish off with, as a conclusion, I'm just going to just highlight again the major things that I talked about, and that is important that you have to either revise again later on, or just keep in mind that that is the things that you will have to look at when you interpret your NMR profiles, especially for NMR metabolomics. So the first thing is that you have spin on your atoms, and that is important to be able to get a profile, and that you have a small magnetic field created by your charges, and that if you put your atoms then in a magnetic field, you can have a transfer of energy levels, or some of that energy is transferred. If you transfer the energy, it can go to a higher level. When it release or emit that energy, it gives you then a release of frequencies, which is then detected, which is then giving your peaks on your analysis. So you two important things that you look at is your chemical shift. So that is important, and that is where it is on your, okay, so your chemical shift, and then look at where on this axis it is. So if it's upfield, if it's de-shielded, then it will be around this area, and if it's shielded, de-shielded, will be downfield, and that gives you an idea of how your hydrogens look like, or the environment of your hydrogens. So that is the chemical shift. The splitting pattern is where your peak is split into different patterns, and that tells you the hydrogens right next to your hydrogen that you are looking at. We then also looked at the coupling constant that you can use later on to determine if it's a triplet or if it's different peaks, especially if we have an overlap of peaks in a complex sample. A nice feature of your NMR is that it's quantitative, so you can get an idea of your concentration of your compounds in there. And a drawback in many cases of your NMR is that it's not sensitive, so you might lose some of your information. But because of the nice application of NMR, and that it's not influenced by a lot of a lot of factors around you when you run your samples, the application of NMR is very nice, especially your concentration that you can find. So there are many advantages to NMR as well as disadvantages, as in any case. So if you have an NMR available, then it's also sometimes that we prefer that you use different analytical techniques. So you can use a LC-MS or a GC-MS with your NMR. Where you have a drawback on your NMR, you can then fill in the gaps with some of your other samples. But yes, this is then the basics of the NMR, so that you know what you see and what you will see on your analysis, and how you will interpret it, and how you will get to answers at the end.
Awesome. Thank you so much, Prof. Prinsloo. That was a great presentation. So as Prof. Prinsloo mentioned, we're just covering the basics. It's obviously a lot more complicated than it is. So you guys can please read more about it if you're interested. There's lots of reviews papers out there on NMR metabolomics. So as I set up my presentation, I just want to thank this MSA communication subcommittee. There was Chanel, Anza, and Kekaletso. You three ladies are awesome for communicating with everyone. Thank you so much. And also to Malati for being available to be a host for this. Your help is always appreciated. Thanks so much. And I'm just going to jump into my one. So I'm going to do some overlap for what Prof. Prinsloo talked about. The interpretation, because as he said, is very important. So everyone can see my screen, yes. To make sure.
Yes, you can see that.
Okay, awesome. Okay, so I'm going to talk a little bit on the practical side, as well as what Prof. Prinsloo mentioned. So very briefly, very simplified NMR metabolomics experiment. Okay, you get your sample preparation. We'll go a little bit more in detail of that. NMR could be quick, 8 minutes up to 3 hours, and how you process the data dependent on your biological question. Are you targeted or untargeted? So sample preparation. Okay, so your sampling is always important. That's in your experimental design. Are you looking at plants, fruits, pharmaceuticals, animals, bacterial cell cultures? In my case, in human metabolomics, we do a lot of biofluids or CSF, cerebrospinal fluids, serum, urine, whatever it is. The great thing about NMR is you can't analyze it as possible. You can't really do that with any sort of chromatography because they'll just clog up your column. You can do a direct MS injection in some cases, direct fusion injection, but that's one of the classic points that I love at NMR. You can analyze as is, but if your sample is too complicated or you want to simplify it or concentrate it, you can do a preparation such as the SPE, solid phase extraction, in which case then you use solvents to clean the sample, and then you have to then dry the sample and then reconstitute it in water. But ultimately, the samples that we usually analyze are polar samples, so they're reconstituted in water. If you want to look at inorganic, then you have to redissolve it in an inorganic solvent, or an organic solvent, should I say, organic solvents such as methanol, chloroform, or things like that. But most of what we do is polar-based. So once you have your sample prepared like this from your sampling, from there on, it's very simple for sample prep for NMR. So this is where the sample prep starts. You centrifuge, 12,000 Gs for 5 minutes is around the standard. You collect the supernatant, and you add that to an epi that contains a small amount of buffer solution. So approximately 10% is the usual amount. So 90% sample, 10% buffer. This is usually a phosphate buffer. Buffer is very important to control the pH. We need to maintain a pH to have repeatability and also to apply the spectral databases that we have. And this buffer usually contains D2O, deuterium oxide, and the NMR internal standard. I'll come in a little bit more detail in this shortly. We do one more centrifugation step because when you change the pH, sometimes some of the particles precipitate in the solution. You just want to remove those to a nice clear solution. And you transfer that supernatant into an NMR tube. So that's the great thing about NMR is all you really do is stabilize it with a buffer and you remove all the large particles and you put it into an NMR tube and you analyze it without interaction with the machine. It stays in the tube, stays intact, and yeah, simplified sample preparation. The internal standard that we use, so Prof. Prinsloo mentioned the TSP. That's one of the more common standards that we use, trimethyl silopropanoic acid. As you can see, it has this structure. Another one option is the DSS, a little bit more complicated, but the same concept as TSP. Why do we use these two internal standards? One, we use it to set at 0.00. So our internal standard, the NMR internal standard, will always be at 0.00. This is for referencing in terms of chemical shifts. Everything will be in relation to the internal standard. As long as you use the same pH and you reference everything at 0.00 for your internal standard lines, everything should be repeatable. That is the way the repeatability lies in NMR. And if you know the amounts of TSP that you're adding to the sample, you can use it for absolute concentration to make everything relative to TSP and to account for the concentration of TSP, and then you get absolute concentration. That's one of the biggest parts of NMR, is excellent, accurate, absolute concentration. So loading the NMR tube, the practical part. So this is our baby in Northwest University, a 500 machine. It's pretty much a large magnet, as Prof. Prinsloo explained. And there is the carousel. Okay, so first of all, we take the NMR tube, we put it into a spinner. So spinner, by this name, it does spin. So in chemistry, they use spinning techniques to analyze samples, but we just use it as a sample holder. So there is one form, just a normal 5mm Gerhard tube. You also get a Shayne tube, a different type, but the same concept applies. Your sample will lie at the bottom of the tube, and you need to have a certain area that must be covered. And this is where the area that gets analyzed. So it does not come in contact with the sample, it just uses magnetic field and radio frequency to analyze the area. So you need to make sure you're within that area. So we load it into the carousel over there. And our carousel over here, this machine holds about 60 samples at a time. It gets loaded into the machine. So here is a cutaway into the machine. It's loaded into the machine, sits in the NMR probe, and that's where the magnet lies. We get magnetic fields, the signal gets applied there at the coils, that's where we receive the signal, as Prof. Prinsloo explained, and we get an NMR signal. So that is the loading of the NMR tube into NMR analysis. And some points to always look at: why do we sell? Prof. Prinsloo and myself, we are NMR people, and we are like salesmen that we want to sell NMR. And what are the advantages of NMR versus MS? So it remains intact in the NMR tube, can be analyzed multiple times in the same tube on the NMR, and can be recollected for future NMR analysis, but also for future MS work. You can re-extract it and use it for GCMS or LCMS. It's highly reproducible results. So we always sell that concept. It's very repeatable, highly reproducible, because it's simple sample prep. Also, the NMR is calibrated per sample. Every sample goes through a lock, tune, shim, and pulse, meaning the whole system is recalibrated per sample. So it is a very repeatable, very reproducible method. It's also very open, in that it looks at the global view of the metabolite. It looks at all metabolite processes. It's not biased towards organic acids, amino acids. It looks at everything: carbohydrates, your nucleotides, volatile things, everything that you can see in NMR in high abundance, you'll be able to see. The major problem is that it is less sensitive than MS. And of course, quantitative is one thing we're both trying to sell across our presentations: accurate, absolute concentrations. Some other points, it's very pH sensitive, so you have to control the pH. That's why we have a phosphate buffer. So we usually operate around a pH 7. That is around the area that we focus on. You can choose different pHs, but then your spectral library database must be at that same pH in order for you to identify components there or metabolites. The whole hardware of the machine controls the temperature. So if you operate at a room temperature probe, the temperature always remains at 300 Kelvin. The hardware, the actual machine, controls the temperature. So temperature is no longer a problem in our labs anymore. It used to be in the past. Temperature would fluctuate, but no longer. It is well controlled. And if you lower the temperature down to a cryogen level, with the cryoprobes, you can increase the resolution and sensitivity, in which case it's been controlled at, say, 4 degrees Celsius. Most of the samples we analyze are in water. So water peak is a big problem, and it has to be suppressed. So the water peak is suppressed, most commonly irradiating the sample. We apply energy to the sample in the form of a program called NOSI-1D. Apply this radiation that suppresses. We cannot remove the water completely, but we can suppress the signal to almost nothing. And there are other programs, pulse programs, when you run the samples like CPMG, that actually help with this as well. And as was mentioned by Prof. Prinsloo, if you increase the number of scans, you increase resolution up to a point. So the 500 megahertz We can go up to 128, 256 scans. Up to about there, beyond there, you won't get any more increased resolution. But the higher the magnet, the more scans you do, the increased resolution. And you got to look at what your project is trying to do. If you want to look at lots of samples, you want to have high throughput, you do a quick metabolomics run. So you do a few number of scans with reasonable amount of resolution to go 200, 300, 400 samples. But if you have 10, 20 samples and you want to get excellent resolution, you can spend more time. So say 1 hour of say 300 scans and some two-dimensional analysis, and you can focus more on the resolution. So it's a give and take for each side. But what is your objective? You've got to choose which one to choose. So just to give some examples, on our side, we do a lot of zoology projects and human projects. Prof. Prinsloo showed you an example of a plant sample, and we played around a lot of zoology projects. So you have some interesting ones to sell. We looked at the honeybee specifically. So we homogenized the honeybee and analyzed it, and this is what it looks like. A spectrum of the metabolome of the honeybee. We also looked at the honey, which is another metabolome all itself. Scorpion, the same concept. We homogenized it, analyzed it, and this is what a scorpion is made out of. This one, the frog, we didn't squash the frog in this case, but we did apply small electrodes to it to apply a small electric field to stimulate the secretion of mucus, and we just scrape that off. And then we put it into an NMR tube and we analyzed it, and that's what it looks like. And it's, yeah, all these samples, and it gives very different spectra. And we trying to identify is a difficult thing because there aren't many databases with frog mucus. So those are interesting projects. And I'm going to reiterate a little bit on what Prof. Prinsloo mentioned on the interpreting spectra. So this is very important, how to interpret the spectra. So having a basic understanding of the theory is good. The practical part is good. But also when we get the spectra, what are we looking at? So when you look at papers, what do we see when we look at the spectra? And what we concentrate on is specifically that we look at hydrogens linked to carbons. So we ignore the OHs, NHs, like any of those type of Hs, we ignore them and we focus on the CHs. And the chemical shift, multiplicity, signal-to-intensity ratio. So Prof. Prinsloo mentioned chemical shift, multiplicity, which you call spin-spin coupling is another term for it. Signal-intensity ratio is another point I want to raise up. So I'll go through this quickly, just to reiterate what Prof. Prinsloo said. So the chemical shift is basically all your peaks in reference to your internal standard. So relative to your internal standard and magnetic field. It is basically where the peak occurs in the spectrum. It's like the GPS of the NMR, where it occurs in the spectrum. And what's very important to understand is the neighboring environment affects positioning, as Prof. Prinsloo showed. So he mentioned upfield, downfield. That's very important. Upfield is as close to one as 0 as possible. Downfield, it's further down, as you can see like that. So I have a little fictitious compound over here that I'd like to use in my presentations, and to show that CH3 and CH3, two terminal CH3s, they will not lie on top of each other, even though they're both CH3s. We have to then look at the environment. This environment here has a double O over there, and this environment over here has an O and a double O further away. And what I like to look at is the electronegativity of the environment. So Prof. Prinsloo mentioned shielding and deshielding. So shielded compounds, such as your CH3s, that lie very close to the 0, they have very low electronegative environment. So electronegative meaning oxygen draws all the double bond oxygen draws the electrons away from these compounds, these hydrogens. So the more electrons are drawn away, the higher electronegativity of the environment, the more deshielded the CH3 becomes. And this double bond oxygen over here has a higher electronegativity than the single bond oxygen, and this one is a little bit too far away. So in this case, the CH3 here lies further downstream than this CH3. And I'll just use this example over here as 111, which is not the actual case, we'll do multiplicity now. And this other one here, the CH, it's more simple, but also in a more complicated environment. It has a nitrogen nearby, and it has other environments as well. So it lies further downstream. So this is just to reiterate what Prof. Prinsloo said, that the positioning is very important. Chemical shift is very important when understanding NMR. And each compound, each metabolite has a very unique fingerprint. Even the cis and trans forms of the same compound have different conformational structures, so they have different positions in NMR. So when I talk about electronegativity, I'll look at this is my version of Prof. Prinsloo's map. And I specifically look at sulfur, nitrogen, and oxygen. Those are the three that we look at metabolites. And electronegativity, other little equation, yeah, sulfur being the least electronegative, nitrogen is the more electronegative, oxygen being more electronegative, and the alkenes, double bonds oxygens and double bonds carbons are the most electronegative. And you can see that in this map over here. The more electronegative, the more de-shielded the compound is. Whereas the least electronegative environment over here are the more shielded environments, more upstream. So Prof. Prinsloo said shielding, de-shielding. I like to look at it as electronegativity, the least electronegative environment to the most electronegative environment. And just to show you here, TSP 0.00. This is a typical urine sample that we look at. We look at a lot of human samples here. And you can never completely remove the water signals. Here is the suppressed water signals. If you don't suppress it, it will just be one giant peak and you wouldn't be able to see anything else. We need to suppress the water signal. Those two are not what we're very focused on. We're focused on all the metabolites in the spectrum. So second point, multiplicity, it's very important. Prof. Prinsloo mentioned the spin-spin coupling, also known as peak splitting. It's a splitting of the peaks, as the name implies. It's the number of peaks that's determined by the number of hydrogens on neighboring carbons. So it's CHs, we always look at CHs. And the general rule of thumb I like to sell in our group is that's the number of neighboring protons plus 1. So this is a very general rule of thumb. There's a lot of exceptions to this. And in chemistry, Prof. Prinsloo explained a lot better than I did that you've got to look at the J coupling and the surrounding environment to understand the splitting. But in very simple terms, what I like to do, and examples I like to give my group, is that when you look at it, you always look at the neighboring environment. So back to our fictitious compound over here, you've got your CH3 and your CH3. These two CH3s, we found out the one was further downstream because of the electronegative environment is slightly higher over there with the double bond versus over here. So it's more de-shielded over here than over here. So further downstream. But both do not have a single peak. This one does because it has no neighboring protons. The protons attached to nitrogen and oxygens we ignore. Whereas this case over here, the CH3, if you look at this, we ignore that hydrogen over there, and you can see it over here as a neighboring hydrogen. And the rule of thumb is number of neighboring protons plus 1. So this CH3 is represented by 1 plus 1 peaks, which is 2 peaks, a duplet. And this very simple CH over here, right in the middle, lies very downstream compared to the rest because of the electronegative environment. And it has only 3 neighboring protons over here. So 3 plus 1 is our rule we use. So it has 4 peaks. So a slightly different approach that I take here, but it's the same concept as Prof. Prinsloo explained. And here are the singlets, duplets, triplets, quartets. It gets very complicated when you get to quintet, sextet, octet, and so on. So beyond quartet, I like to just stop and say anything beyond that is a variable number multiplet, so many peaks. And that can usually be the case when you come to the aromatic regions. Lastly, signal intensity and ratio. I think that's also very important because the CH3 and the CH3, they're both represented by 3 hydrogens. So in the past 2 rules, chemical shift is an environment, electronegative environment, affects the positioning. The multiplicity is the number of neighboring protons, affects the splitting. But in the case of the actual, what we're analyzing, the CH3 and the CH3, this CH3 and this CH3 both have 3 protons. So that area under this single peak is equal to, if it's equal to 3, the area under these 2 peaks, if you add them together, must also equal 3 because these are 3 to 3. These are a ratio. 3 hydrogens, 3 hydrogens. This little guy over here, this little CH over here, only has 1 hydrogen. So if you have to calculate the area underneath there, this will be 1/3 the area of this. So it's always important to look at the ratio of 1 to 3 to 3, 1 hydrogen to 3 hydrogens to 3 hydrogens. So the peak area is proportional to the number of protons. So this is just reiterating what Prof. Prinsloo went through. Coming back to the practical side. So this is a summary of the 3 things I mentioned. But I want to come back to the practical side of looking at examples. Examples is always how you apply what you've learned of what we've shown you. And I like to use this example because this is a compound over here that all students love to engage in the weekend. This is ethanol. Ethanol is a nice linear structure. Prof. Prinsloo had dichloroethane. No, it was just chloroethane. Hey, chloroethane. We've replaced it with an OH. This is a little bit less electronegative, but it'll give the same spectrum because there's a CH3 and a CH2. So we apply what we learned with the theory part, the background part. We've got 3 hydrogens lying far away from the oxygen. So this is the most shielded or the least electronegative environment. So it lies further upstream. So this is the map I'll bring up again, and you can see the CH3 lies over there, little methyl group. So we expect it to lie somewhere over there. The CH2, okay, it has oxygen next to it. It's a more electronegative environment. So we expect anything with the CH2 with the oxygen next to it, somewhere to lie in this environment. So CH3 in this environment, CH2 in this environment. That's a chemical shift, the first point. The second point is a multiplicity. The CH3 over here has 2 neighboring protons. So this CH3 should represent about 2 plus 1. Splitting some of the neighboring protons plus 1 should be 3 peaks. The CH2 over here, we ignore the OHs and NHs. It has 3 neighboring protons. So the 3 neighboring protons plus 1 should give 4 peaks over here. So here we are, the location is there for the CH3, and that is a triplet, and our CH2 is a quartet. And the ratio, this is 2/3 the area of this. If you had to add all these peaks areas together, this will equal 3. Add all these peaks together, this will equal 2. So the third point, signal intensity and ratio. So this is a fairly simple compound. This is a more of a difficult compound, but very important to us because we do a lot of human samples and urine samples, and creatinine is a very important component in urine samples because we make all concentrations relative to creatinine. Creatinine has a CH3 and a CH2 as the one we looked at previously, but the environment is a lot different. The environment changes the spectrum. So this is a much more electronegative environment. The CH3 has a neighboring nitrogen. This CH2 has a neighboring double bond oxygen. So if you bring up our little spectrum here, so this is actual urine sample over here. These 2 peaks here represent creatinine. This guy over here, this is the CH3. If you bring up our little map, you can see the little nitrogen next to it. It lies in the nitrogen region, which is approximately 3 there. This is at pH 7. And then the CH2, which lies over here, is where the oxygen further downstream. So if it wasn't a double bond, it was just a single bond, or maybe lies a little more upstream, but the double bond oxygen means it lies further downstream, lies over there, about 4 in the region over there. And both of them have no neighboring protons. So look at the CH3 here, this immediate area here, we ignore this H over here, no neighboring protons. This one's a little bit too far away, but 1, 2, 3, a little bit too far away in this case, because the conformational change, the formation of the structure means this is too far away to affect the position here. So this is a singlet, it's 0 plus 1. Same as this guy over here, 0 plus 1, both of them are singlets, 1 peak. And this is an area of 2 representing CH2, and this is the area of 3 representing a CH3. So I hope with all these examples, starting to put it all together, that each chemical compound has a unique fingerprint. So moving forward, what are the applications? Applications are the most important part, in my opinion. You can have a targeted application. So we are focused on human samples and we want to diagnose people. for certain metabolic disorders. So if we suspect a person has this disorder, this is a metabolic disorder, there's an accumulation of this compound, 3-methylbutyronic acid, specifically this trans and cis form. This is not a natural compound to have in your urine. So you have these two other components here, 3-hydroxyvaleric acid. You can target the spectrum. If you overlay pure compound spectra and you confirm the identity of these compounds, and you say these are present and these are present and these are present at this concentration, this will give you a diagnosis that this person has this disease based upon these metabolites being present in the urine. So this can be applied across all different types of metabolic disorders, and there are a lot of them. Some of them we can't identify if we can't see that the compound in low, if it's in low abundance. That's the biggest difficulty of NMR. If it's very low abundance, we can't see it. If it's in high abundance, in this case, these two cis and trans forms of these metabolites, which shouldn't be in urine, are now present in urine. If they are present, this is a very good indication for a diagnosis for this disease. And if we use a 2D to confirm it even further, then we have an almost 100% confidence. I won't say 100%, almost 100%. And we're very certain that this is specifically this disease. So that's the application in a targeted sense for targeted metabolomics. Untargeted is another story. So application, we're here for metabolomics. We look at disease and control. And we look at either targeted profiling. So if we know what you want to look for, you can then look at those specific metabolites. If you're untargeted, you don't know what you're looking for. Untargeted, then you got to look at everything. And everything means you got to break up the spectra into digestible bits. So you can't look at everything at once. You got to break it up. So binning just means dividing the spectra into equal amounts or variable amounts in some cases. So yeah, it's binning. And you can see the bottom here. This is the ppm, 3.222426. So this is 0.02 binning. So for every 0.02 interval, we have a bin. So this is one bin, one bin, one bin, one bin. And these 4 bins contain spectral information. These 4 bins over here, 1, 2, 3, 4, are noise. We can ignore those. And these 4 over here have spectral density. We give this to as a statistician to go through all the statistics to identify the regions. What regions? So if bin 2, bin 4, and bin 8 are the major regions that differ between your experimental group and your control group, then you can go there and you can identify what is present there. And you can do a metabolic interpretation. So you need to create a big table of bins. Some of them you can identify, some of them you can't, they'll be unknown. You have a large data set, you give it to a stats person. The stats person then does all the statistical analysis. So the PCA, looking at the loading plots, trying to validate it to do predictions. And then you come to a list of very important metabolites to compare versus patients. So you have different control groups maybe, or in this case, we had a collection compared to 2 controls, and these are all the metabolites we found were increased and some of them were decreased based upon the bins that were identified on the stats. And then we take these bins and we apply our biochemical knowledge to it. And from there, we formulate hypotheses. So untargeted metabolomics is hypothesis generation. That is the biggest selling point of untargeted metabolomics. We take a discovery and we try and put it all together. And in this case, we put it together in an astrocyte microglia lactate shuttle. So in future journal clubs, we are hoping to see a lot more application of this, so targeted, untargeted. But for now, this is just a brief introduction into this world of NMR metabolomics, targeted and untargeted metabolomics, the foundation of what we discussed. And I'm sure lots of you have questions on this. I'm not sure if we have time for questions, but we do have a forum where I encourage everyone to go there and register, and you can introduce yourself, communicate. We want to try and get more of the communication going on with the MSA community. You can post your questions there. We're going to have another session at the end of April, our 4th session, in which we're going to have a Q&A with all our speakers, myself, Prof. Prinsloo. Next week, we'll have Prof. Edwin and Prof. Fidel doing the GCMS and LCMS. Prof. Fidel from UJ, Prof. Edwin is from Venda University, and they'll join us on the last session of April for Q&A, hopefully. So any questions, put them here, and we'll try and compile them together, and we'll give them to the specialists and see if they have answers for you guys. And then on the third session of this month, the communications committee will send out an email on that explaining what's going to happen. We're going to have a trial run with 2 students presenting their projects. And that is from me, my contribution. So please communicate with each other, communicate with us. That is what we're here for now, is to have a constant communication. So I hope everyone took something away from this. Thank you very much to Prof. Prinsloo for joining us and giving the background that you gave. And yes, Malati, I don't know if you want to add anything to close off, or the subcommittee, communication subcommittee. No. Okay. So this is recorded. So we'll try and create a Google Drive to keep a record of everything. If anyone wants to request access to it, we can arrange that. But yeah, please come to the forum. We're open to everyone, and share your projects with us. We want to give feedback. We want everyone to be involved with each other. And please join us again next week for GCMS, LCMS. Prof. Fidel and Prof. Edwin will be sharing their experiences and knowledge with us.